Hello, my name is Rachel Snyder, and the purpose of this video is to talk about a new technique that could revolutionize the way we think about genetics. Before I talk about CRISPR, I think it's necessary to talk a little bit about what we know about genetics up until this point. Introductory biology courses teach about Gregor Mendel and his pea plant experiments during the mid-1800s, but it was almost 50 years before anyone paid him any attention. The belief up until this point was that genetic material from the father and from the mother were blended to form an offspring. Due to luck or the grace of God, he was a monk after all, Mendel's pea plants were an ideal model organism because they had visible traits that were easily manipulated. Post-Mendelian genetics moved slowly until the Hershey-Chase era during the 1950s, when DNA was confirmed as the molecule of inheritance over proteins. Now, of course, we have Rosalind Franklin and several others that deserve credit for major discoveries in this time frame. I say all this to make the point that within the lifetime of the baby boomers, the field of genetics has gone from the discovery of the double helix to new technology that allows DNA to be edited. So where did CRISPR come from? Clustered, regularly interspaced short palindromic repeats, or CRISPRs, are sequences of DNA that function to take up genetic material from pathogenic substances that attack bacteria and archaea. It acts as a defense system. It allows for heritable immunity because the sequences are embedded within the host genome. These repeats were discovered in the late 80s and early 90s by Francisco Mochka, but it wasn't until the early 2000s they were linked to antiviral defense. The bacteria are able to use this enzyme, which has since been named Cas, to add the new sequences to their genome. Cas9, which is the one that is most commonly discussed, was discovered by Alexander Baudelin. He is from the French National Institute. He noticed that the Cas9 protein was a little different than the other ones because it had a nuclease activity. The protospacer adjacent motif, or PAM, is another sequence that follows the spacers in between these other sequences that act as binding recognition. The latest research is focused on how this technique can be used in a laboratory setting to edit specific sections of DNA. There's an argument about who actually figured this out. Jennifer Doudna and Emmanuel Charpentier showed that the editing aspect of this could be incorporated. However, someone else beat them to the patent office, but the scientific community still credits the two of them for their discovery. So how does it work? As mentioned earlier, CRISPRs are sequences of DNA and bacteria that are taken up from pathogenic substances and provide a way for bacteria to recognize the pathogen the next time. It's a similar concept as our immunity, just with completely different mechanisms. We use antibodies created because of previous infections or vaccines. They use pieces of DNA. The interesting aspect of this is that the bacteria are able to encode this genetic material in their DNA, so the immunity is heritable. The crossover to applications in multicellular organisms is where we can use this to edit genomes rather than immunity. It uses short sequences of RNA. These are often referred to as CRISPR RNAs or cRNAs, and then they in turn code for guide RNA or gRNA. gRNA is complementary to the target DNA. Then Cas comes in, shuts off the gene by cutting it, and as mentioned earlier, Cas9 has the nuclease activity. An important note here is that it can cut both strands of DNA at a very precise location. gRNA is much easier and cheaper to produce than the alternatives, and those are typically transcription activator like effector nucleases or zinc finger nucleases. Research has now shown the ability of this to not only cut and delete, but also to paste. This would allow for the ability to fix not only insertion mutations, but deletions, any sort of frame shift, and nonsense mutations as well. The system can be used to target several genes at once, which is an important trait that sets it apart from other gene editing techniques that can only do one. So, modern experiments, recent discoveries, why does this matter? The most obvious uses of this technology deal with genetic diseases. Cystic fibrosis is commonly caused by a mutation in the cystic fibrosis transmembrane conductase regulator in the 508th position with a deletion of 3 nucleotides that cause phenylalanine to be deleted. Sickle cell disease is caused by a mutation in the HBB gene. A normal beta globin subunit is replaced with hemoglobin S. These are both examples of genetic diseases that are caused by only a few base changes. Both could be eradicated by the perfection of this technology. Overall, CRISPR is not considered to be worked out in humans, but I will discuss one example of that later on. In 2018, a team from Paris published a study entitled Potentials of CRISPR in Liver Research and Therapy. They focused on genetic diseases, but also pathologies induced by viruses. They begin by discussing the Creo-Lex system, which is not something that I've had any dealings with in my lab experience. It uses Cre recombinase to catalyze sequence-specific recombination. The figure compares the use of Creo-Lex with CRISPR and Creo-Lex in addition to CRISPR. The problem with this technique was not that it was ineffective, but rather the long-term effects on the organism being used. In this particular study, as well as many others, mice were the model organism, and they required constant breeding and transgenesis because the technique was prone to kill the mice after repeated use. This issue could be avoided entirely by using CRISPR. So they induce a loss of function in a common tumor suppressor genes that cause liver cancer. The results yielded all of the mice dying within 4 months due to tumor development. They then sequenced samples in order to determine which specific mutations led to the highest development of tumors. This experiment gives valuable information to the clinical research regarding therapy for things such as liver cancer. Each study done that furthers this technique allows the scientific community to be one step closer to clinical applications of this revolutionary idea. There are many studies published just in the last few years that have progressed CRISPR from an idea to a usable technique. Several that I've seen lately, such as this one about liver therapy, are focusing on treatment rather than prevention. This is important because preventing genetic disease would be ideal, but what about the people who already have CF or those with liver cancer right now? So something else to think about are the ethics of this technology. The idea of CRISPR is amazing. If I was told I was going to pass on a high genetic likelihood for my child to develop breast cancer, I feel like I would do almost anything to reduce that chance. However, this technology is not black and white. It's not something that's 100% reliable because we still have so many gaps in our knowledge about genetic material. If you think about it, I mean, we've only known that DNA was the molecule of inheritance for about 70 years. With things such as alternative splicing, one gene can code for many different products. Our bodies produce about 100,000 proteins, and old estimates said we had about 100,000 genes. There'd be one gene per protein. But recent estimates place that number around 30,000. Whenever we set up a reaction in the lab, we assume that there's no way to get 100% of the expected yield. It's a problem I've worked on many times for a lab report. It's why we use so many plasmids, so many bacteria, to hope that just enough of them transform so we can culture them. We cannot assume that 100% of what we will do will work in any experiment, much less something that's this sensitive. A team in China published their experiment in November of 2018. They claimed that they have used CRISPR to create twin girls that are resistant to HIV. The lead scientist, He Jianju, is under a great deal of scrutiny because he did not ask for forgiveness or permission. Rather, he said that he did it and he would do it again. He's quoted saying that all they did was add a little protein and some information. From a scientific perspective, I mean, that is all he did. But from an ethical perspective, how does he expect to test the theory that his twins are resistant? Are they going to expose these children to HIV? The HIV resistance has to do with where HIV attaches to CD4 cells. There's a protein on the surface, and without this protein, the virus cannot effectively attach to the cell. If it worked, I mean, we would have a way to prevent the spread of such a terrible disease. But how can we tell that that's the only piece of information that was changed? How can we assure that our changes are only the intended ones? There's a little bit of food for thought. What are ethical issues that it can create? We can take away bad, but we also could add good. What about enhancing ability to be athletic or otherwise? You know, what in 50 years problems are we going to have because of this? So overall, CRISPR is a gene editing tool that has made leaps and bounds in the field of genetics. The scientific community has learned a great deal about specific genes through this technology. I had a professor say the best way to tell which light switch belongs to which light was to turn it off. And the same that applies to genes. CRISPR allows us to accurately do that. We can turn them on, we can tell exactly what they do by the effects that it causes. Research now focuses on removing genetic disease, therapy for existing disease, and so many more things. Within our lifetime, CRISPR could become a common phrase with unlimited potential in medicine, genetics, microbiology, and so much more. Thank you.